Hello, everyone. Thank you for joining us today for our webinar, Designing Success in Your Fresh Cut Fruit and Vegetable Process. This webinar will be hosted by Jeff Brandenburg of the QFresh Lab and Joelle Mosso of Eurofins. I'm Amy Knock, and I'll be moderating this webinar today. Before we begin, I'll let you know more about how this webinar will run. This webinar is being recorded and the slides and recording will be available for you within 3 business days. A short Q&A session will follow the presentation today to answer viewer-submitted questions. During the webinar, you can submit questions you have using the webinar sidebar menu. Select the Questions tab, type in your question, then hit the Enter key on your keyboard. Remember, you can submit questions anytime throughout the webinar. Now at this point, I'm going to turn it over to Joelle. Joelle, you can go ahead and begin your presentation.
All right, well, thank you, Amy. And thank you everybody for joining today. Today we're going to talk about how you can avoid costly errors in your fresh cut process and basically design success from the start. And with that, I'll let Jeff introduce QFresh Laboratories and I'll follow with Eurofins.
So good morning, everybody. Thank you, Joelle and Amy. This is a full service and analytical testing laboratory supporting the fresh produce industry. And our specialty is shelf life testing and optimization, modified atmosphere packaging design, food safety planning review and audits, and analytical services around packaging and packaging properties, including oxygen transmission rates, sustainability, integrity, all things to do with packaging. So we really are unique in the industry in that we are a quality and shelf life and packaging testing firm. Back to you, Joelle.
All right. So for those of you who aren't aware about Eurofins, we're actually the world's largest testing company with over 800 different locations in over 50 countries. And we have a whole host of employees and more importantly, analytical tests. So if there's a test that can be done commercially or that needs to be developed commercially, Eurofins is basically your service provider. We pride ourselves on integrity as well as providing customized support and transparency of how we get to your test results. But I think more importantly, if we put into the context of what we're talking about today, is that we provide a whole host of systems to help you with product development as well as sensory. We have a certification body to do food safety audits as well as consulting. And then finally, any kind of process and project validation. So Eurofins really is we're an international company here to actually serve you and find ways to provide added value to make your jobs easier in what you do. So Eurofins and QFresh Lab have joined together in a strategic partnership predominantly to fill a void in which Eurofins as well as QFresh we're not able to provide, is that whole continuum from seed to consumer experience. And that effectively starts with seed development and the plant health itself in the field. But then when it gets cut, it becomes a physically different project, and that expertise that Jeff just mentioned comes in very much into play. So we now have expertise in fresh cut physiology, well with the whole host of testing and analytical services that come along with Eurofins. Additionally, we have experts in quality and food safety, microbiological testing, as well as nutritional fate and contaminants, as Jeff mentioned, the modified atmosphere packaging, and sensory studies and validations. So effectively together, Eurofins and QFresh Labs have been able to serve you from seed to salad plate, if you're thinking of it in terms of fresh cut salads. But obviously, there's a whole host of other variants of fresh cut products that this is now going to serve. So we're very excited about this partnership because it adds added value and ease for our customers, as well as fill the gap into knowledge bases that actually interplay quite a bit, as you'll see from this presentation. And so now we can serve you even better in that capacity. So that's our partnership. So starting in terms of seed development and health, Eurofins has a whole business called Eurofins Diagnostics that's working on developing better and more vigorous seed varieties. So we're here along with the growers and the seed developers to address plant pathogens that might be in the field that would ruin your yield and obviously your shelf life if you have a plant that is not healthy and vigorous when you bring it into your processing plant. It's certainly not going to get better later on in the process. Biodiagnostics uses cultural as well as plant trials and genomic testing to be able to screen and diagnose these issues. And so we work alongside seed developers as well as processors who are trying to select for different seed varieties to make sure that they're going to be the best suitability for the season, for the product, and obviously for the shelf life. So there's just some examples of some of the things that they do: bacterial fruit blotch testing, bacteriology, mycology. We do downy mildew analysis and different bacterial wilt and fusariums, and obviously the bacteria like Pseudomonas and stuff that can plague your shelf life products. So the seed development and health, Eurofins has a whole host of experts that are here to help you in that respect. And then where does it go into the variety fit? So I'll leave that to Jeff.
So from the QFresh perspective, and as Joelle mentioned, what makes this relationship very, very exciting for us as well is that both Eurofins and QFresh are looking at the same part of the supply chain, but from different angles, benefit to our customers and all the way up to the consumers. So when we look at seed variety, we're looking at affecting shelf life and quality in outdoor or even indoor grown produce cannot always be controlled. Seed variety can be controlled, and by testing that seed variety, which is one of the services that we provide, by testing that seed variety commercialization, we can assist you in learning what the quality traits are and how that seed variety how it will impact the modified atmosphere package, how it will impact product handling. And then the seed grower can actually begin to breed traits that can impact shelf life, impact the entire distribution channel. So we feel that this is a very important part of the services, and we want people to understand that don't just think of it at the tail end and okay, here we've got the grown product, we've packed it, now we're going to start to think about shelf life, but think of it much, much earlier on. So when we look at in the next we do, you can see, and go ahead and switch to the next slide, Joelle. You can see we're looking from an analytical chemistry perspective and also a seed variety perspective, and colors and textures and sizing and consistency. Do we need modified atmosphere packaging or not? And we'll get into what that actually means in a few. By having all of these testing procedures under one roof, we can look at a successful seed variety trial, at those seed varieties for different applications within the fresh produce industry. So let's take a minute and talk about and why is fresh produce different. One of the fascinating things about working in the produce industry from a pack point is fresh produce is alive. It breathes. It consumes oxygen. It gives off carbon dioxide after harvest. And it's sick. It releases heat. And then, of course, as we all know, it can even die. So when we are involved in looking at how to optimize shelf life, how do we keep food safe, we have to understand that unlike a lot of other food kinds, Um, and that causes the respiration rate of the produce to go up. And the warmer it is, just like humans, the harder we breathe, the faster we breathe. The cooler the temperature, the less that we breathe. So when we're talking about shelf life, quality, shelf life extension, quality optimization, we're always focused on the temperature at which the produce is being exposed out the throughout the distribution channel. So respiration rate is high respiring leaves deplete pigments and nutrients faster, and then can cause yellowing and decay. Seed varieties can influence those kinds of shelf life. They can influence the respiration rate levels and how the produce will react under certain environmental conditions. So it's important to understand that we have a living, breathing product. And we are, once it's harvested, begins to consume oxygen, begins to give off CO2, and you're working with those physiological properties to optimize the quality and the shelf life of the product. Back to you, Joelle.
All right. So Jeff just explained a lot of the complexities of what happens to a plant once it's cut and it changes its metabolic rate. I think the thing that we often maybe don't think about is that we're also changing the microenvironments that are happening on that plant surface as well. So to make an already challenging situation harder, we're now adding a whole nother ecosystem that we need to think about that has changed and shifted when we've brought that package or that product out of the field. So produce items commonly harbor somewhere between 10 to the 6th, so a lot of bacteria, 10 to the 6th to 10 to the 8th, so millions and millions upon bacteria per gram on a leaf or a plant surface. And generally, when you have a healthy living plant, they have their own immune system. They have their own way to balance those pathogens as well as just normal organisms, somewhat similar to humans that have bacteria all over our hands, but we don't get sick. The issue is once you cut those products, that suddenly that shifts, that stability shifts, and we now have to think about how those microenvironments are also changing within your package. So part of the unique thing about the partnership between QFresh and Eurofins is that, as Jeff had mentioned before, is that you need to think about all of these variables in advance, not because, certainly because they're all going to impact each other in the finished product at the end. But the reason you really need to think about them in advance is that they're all happening concurrently. And if you forget to consider the variables about bacteria and your commensal organisms that might be on the outside of plant surfaces, is that you may design a perfect seed variety and you may understand exactly how your plant is going to respire. And then you may be completely foiled by the bacteria and viruses that suddenly are on the plant surface that just cause your shelf life to tank. So your microenvironments evolve, and they evolve at different stages in your process, partially because you're changing the product at every stage. And every time that surface changes or that system changes, so do the microenvironments that have these microflora. Some of the things to think about in respect to that, our plant varieties, so that seed development to begin with. Some are more susceptible or more rigorous than others. The season, depending upon the temperatures as well as water saturation, you can have different environments as well. Also, just weather and different practices that might happen. So did you just recently fertilize, or did you apply some other crop input? Well, that's going to change the surface and microflora that's on your product. So all of these things are different variables that will impact the micro environment on top of your surface and ultimately impact your shelf life at the end. So the key to fresh cut shelf life actually starts far before you get it even near the package. Your plant genetics themselves, how rigorous are they? How susceptible are they to different plant pathogens? And then obviously what microflora live on them so that when you cut them, they're suddenly going to be part of effectively your ingredient. You need to understand the plant physiology, as Jeff mentioned. Temperature and respiration rates will significantly impact not just your plant, but also what's living on the surface of your product. And then certainly do your quality and pathogen testing, because once something's destabilized, you have an opportunity for different things to take over. Harvest time, another thing that everyone who's been involved in fresh cut produce knows. There's, the plant itself is in different states at different times of the day. Some of them impact your shelf life in a more favorable fashion. So night, morning, or afternoon, these are all variables that you have to think about prior to even getting to a point of knowing if your product's going to survive in the shelf life that you thought. So following all of that, if you actually can get through all of that, you go into your processing step. And the processing step, the processing has certain pillars. Now, all of these pillars may not come in this linear fashion, but they actually might be something that's a little bit more variable. So maybe you don't have washing and drying within your system. Maybe you have prep or work in progress that's at different stages in your process. So all of these things are things that you need to think about and not necessarily in a linear fashion. So if you start thinking about packaging after you've done an enormous amount of prep and figuring out the seed varieties and how you're going to wash it, or how you're going to dry it, or how you're going to cool it, you may find yourself in a situation where you could never have won because you didn't design everything at the same time. So really being proactive and thinking about each stage and how they impact the other stages is critical in your processing. This will be, prepping, whether you're cutting it, if you're applying sanitizers or some sort of water. If you're not applying water, is it a forced air cooling? What kind of cooling method are you going after? All of these things will change ultimately the physiology as well as the microbial ecology of what's on your product. So like I just mentioned, is that every step is effectively creating a new product for you. And every step then needs to be analyzed to understand how it's impacting what could ultimately be a successful product at the end. So if you're washing something, you're introducing a new microbial opportunity. You're going to be shedding or spreading whatever's on the outside surface to other products as well. You could be introducing new organisms that are capable of living in the wash water. You're going to be introducing chemicals, and that may cause your plant to actually respond differently. And you have to think about that in terms of both sensory, will your consumer taste a chemical, and or will it change the respiration rate that Jeff mentioned? Because all of these will be critical to basically impacting if you can hit the shelf life that your customers and consumers need. As you're processing, there's conveyor belts, there's wash flumes, there's tons of mixing tools that make your product possible. But those are also new surfaces that over time through your production run may actually be growing other microbial environments. And those can effectively then impact the quality of your product at the end. So if we might think about as an example with fresh cut apples, is that, okay, we have 10 to the 8th, 10 to the 9th, cells per gram on the outside of our apple. As we slice them, those surfaces that actually slice the apple now become harborage points for bacteria and yeast and molds, all of which may have impact on your shelf life at the end. So you may be able to actually design a process where you monitor through environmental monitoring non-pathogens and actually just quality and spoilage organisms, and be able to correlate a shorter shelf life with when it was processed in your production run. And that's because over time, those surfaces that are touching your products and those high microbial counts are actually growing counts of their own, and they're effectively inoculating things later on in the process. So when you think about your process in terms of that, as each stage is actually impacting in a positive, negative, or moot point, you can then design where you can put your mitigations and or steps to be able to design exactly what you're looking for in terms of shelf life and performance. So these are all things you should think about. And we often talk about environmental monitoring just with pathogens, but really we should be thinking about, especially in fresh cut produce, is that our environmental monitoring is going to show us what's there and potentially what's going to impact our product's success because we're building up that microflora. And those can be anything from aerobic plate count to lactic acid, bacteria, or yeast and mold. It's somewhat dependent on what kind of product you're looking at, and then certainly what kind of spoilage you often see. And keep in mind, all that can change seasonally because so too does the outside of whatever's growing on your plants. I'll leave it to you, Jeff.
So, so the importance of understanding when you start to put product into a package, it's important to understand, as we both said, the physiological properties, and it's also understand what is the incoming microflora. Because as we're going to talk about, whenever you put produce into a package and you seal that package, the produce is going to begin to consume oxygen and give off carbon dioxide, as is the bacteria. So, and by definition, a modified atmosphere package is any package that doesn't have atmosphere inside, 21% oxygen, 300 parts per million carbon dioxide. So as soon as you seal that package, your produce is going to start consuming oxygen, giving off carbon dioxide. So you've just created a modified atmosphere packaging. You don't have to inject gas in it. You don't have to do anything. By scientific definition, you now have a modified atmosphere package. It may not be correct, it may not be desired, and it's almost guaranteed not optimized, but make no mistake, you have created a modified atmosphere package. So this then begs the question, how do I optimize that package? Next slide, Joelle. So when you optimize the package, as we've said a number of times now, you're looking at the produce physiological requirements. You need to understand and quantify how your produce is reacting to the environment. You need to look at your polymer engineering. Okay, how are we going to make this? Is it going to be a fiberboard tray? Is it going to be a recyclable plastic tray? Is it going to be a bag, a stand-up pouch? And then you need to take into account, how are we going to fill it? Are we doing this manually? Are we putting in a packaging line, packaging equipment? You know, how are we going to get the product inside the package? And then we have to look at the marketing and the branding requirements. What do we want it to look like? What do we want it to feel like? What message are we getting to the consumers, not only about the product, but about our company and or our brand? And then certainly most recently, sustainability, and we'll talk about this in more detail in just a few minutes. Sustainability aspects of the whole product become very, very important. So when you're looking at designing a modified atmosphere package that's going to optimize fresh produce quality, it's critically important to take into account all of these aspects. And as Joelle mentioned earlier, if you start the package design at the 11th hour, you will never optimize the package. The package design process should in fact be done very early on, along with the development of the product itself, of the produce itself. So in the next slide, what we look at is, what are the benefits of modified atmosphere packaging? Why do we want to do it? Do we need to do it? And that depends. So what modified atmosphere packaging does for you, what you want to do with modified atmosphere packaging is you want to reduce the oxygen inside the package and increase the carbon dioxide. But you want to do it in a very controlled manner. By reducing the oxygen inside the package, it slows down the respiration rate of the product. So it begins to put it asleep. It slows down the chlorophyll, the nutrient degradation, and enzymatic activity. And in certain cases that are prone in certain produce, in certain types of produce that are prone to enzymatic or browning reaction, you actually want to get that oxygen level down very quickly. But by reducing the oxygen level, you're slowing down the physiological properties and you're putting the produce to sleep. You're in certain cases where necessary, preventing enzymatic browning reactions. And then by the converse of that, by increasing the CO2, you're impacting and limiting the growth of aerobic microorganisms. So things like Pseudomonas and yeasts and molds, you'll impact by creating a more acidic environment on the surface of the produce, you'll impact or minimize the growth of those materials. So in a sense, all of that, what it does is it begins to allow for a longer shelf life. You slow down the respiration rate of the product, you've minimized bacterial growth, you've minimized enzymatic browning reactions. Now, in the next slide, what we look at, so what we're really trying to do is we're trying to get gases to come in and out of that package in a very controlled manner. And it may be through breathable polymers, it may be through a breathable patch, it may be through microperforations, but we need to make sure we understand how gas is moving in and out of that package. And ultimately, we need to understand what kind of gas concentration is left in that package. So having something like a MOCON or a checkpoint O2 CO2 monitor is critical in understanding what's going on inside that package. Now, as I said earlier, because bacteria also consumes oxygen and gives off carbon dioxide, if you've got a really heavy incoming microflora count, they can actually take over and create the atmosphere within inside that package over and above the produce itself. So here we get back to why it's so important to understand what the incoming load is on bacteria on the produce coming in, and how do we control that. So when we design that package, we're looking at how gases move in and out, and we're attempting to create a specific gas in that package. So, and if we look at the next slide, we can actually see what the actual equation is that's used in order to do that. But what's important here is that this is all based on temperature. So if we determine what the physiological properties are of the produce at a specific temperature, and that temperature is not relevant to what is actually being used throughout the distribution channel, then the packaging will actually be invalid. Because as the temperature increases, the O2 CO2 respiration of the produce increases. We've designed the package to let gases in and out in a given manner at a given rate based on the physiological properties of that produce at a given temperature. Now, all of a sudden, that produce is respiring at a much higher or much lower rate, which means the package is not going to allow the oxygen in and the CO2 out at the rate that it needs to be. So we cannot overstress the importance of temperature as it relates to modified atmosphere packaging design. And there are actually conditions where there's tremendous temperature variation, where modified atmosphere packaging can actually do more harm than good. So when we look at what is that target atmosphere, that's the kind of information that the QFresh Lab has through proprietary measures that we can help, create those optimal packages. What you don't want to, so as I said earlier, we want to get the oxygen level down and we want to increase the carbon dioxide level, but we don't want it to go anaerobic. We don't want we don't want the produce to suck all of the oxygen out of that package so that the produce turns from aerobic metabolism into anaerobic metabolism, because then if you're in anaerobic metabolism, fermentation occurs, and you can get all kinds of ethanols and aldehydes formed, which gives you the off flavors and the off odors. So again, this is why the temperature is so important. We want to reduce the oxygen, but we don't want to dive it down into anaerobic. So if you look at the microbe physiology of modified atmosphere packaging, and getting back all the way in the next slide, all the way back to the seed development, you can begin to understand why it's so important to understand the physiological properties of not only the produce itself, but how the seed impacts it. Because atmospheric changes on the surface of the product due to modified atmosphere packaging is going to change the populations of the bacteria. Plant physiology changes can lead to microbiological changes. Understanding the changes within the package over shelf life will help you to identify where and what your product, where and how your product is spoiling. So we need to understand, and you're getting this reoccurring theme. We need to understand and quantify to the best of our ability all the way back to the seed as to what this product is doing, so that we can design the packaging around the physiological and microbial properties of this product to optimize your shelf life, quality, and even safety. So I want to spend a few minutes kind of talking about sustainability, because this is a huge, becoming a very significant topic in our world today. And sustainability is a very broad term, and there's lots of ways you can attack it. And there is no one right answer. If I just do this, all the problems were solved. Because sustainability includes recyclability, it includes compostable products, both industrial and consumer, and there's a very significant difference between industrial compostable products and consumer compostable products. We're trying to keep products out of landfills. Are we using renewable films, plastics, products? Are we going to use degradable? How do we, when we talk about recyclable, we need to know that different communities recycle at a different rate. So when you're talking about and you want to design a quote sustainable package, that's easy to say, but it becomes much more challenging when you actually want to understand how to do it. And a lot of this goes back to the distribution channel that your package goes through, which may make one sustainable option more advantageous than another sustainable option. In some areas, recyclability, an APET tray, which is commonly recycled throughout North America and really throughout the world, becomes a good sustainable option. In some cases, people don't want plastic looking things at all. Okay, we can go into some kind of a fiberboard product. The challenge with that sometimes is it reduces visibility. So if you look on the next slide, you can see that there are all kinds of different ways to attack the sustainability goal, but there's advantages and disadvantages of each. So like designing a modified atmosphere packaging, like optimizing the produce quality and safety throughout your entire processing chain, harvesting, packing, you need to understand the properties of the produce, what it's going to go through, and how the package is going to react to that. I mean, fundamentally, packaging is designed to keep what's in, keep what's out, provide physical protection, and provide some marketing, some graphics. This is who we are. This is the message that we want to get. So there's all different kinds of ways. And then in fresh produce, of course, we've got this symbiotic relationship between the produce and the package, where we're needing to bring oxygen in and carbon dioxide out. Now, one of the interesting things that's really coming on strong now are biopolymers and a variety of natural polymers that we can use. And these are changing almost daily, and new developments are happening. There are new polymers from food products. People are looking at waste mushrooms, kelp, tomato peels into coatings. Sugarcane now is becoming sugarcane husks. The part that you don't use when you're making sugar are being turned into fibrous trays. Rice husks are being turned into fibrous trays. So there's a tremendous amount of research and development going forward in how to make a sustainable product, how to keep plastics out of the ocean, how to make sure that we treat the environment properly. But as I said before, not one solution fits all. And it's really important to understand, like we've said on everything else, understand your product, understand the distribution channels, understand what it's being used for, what the consumer requirements are. In many cases with fresh produce, consumers want visibility. Well, that causes a challenge. So that needs to be understood, and you work around that. So kind of in summary, if you will, if you look at the next slide, optimally designed packaging plays a critical role. It's not the only thing that you need to be aware of, but it's certainly one of the key things. Customer requirements are very, very important. And based on these customer requirements, creative technologies can be cutting edge and untested for performance and need to be thoroughly understood. And the customer's requirements need to be thoroughly understood so that you pull the right lever for the best choice. You can measure performance at different stages, but results will vary compared to the whole process. So as Joelle said much earlier on, when you're looking at those stages of development, you look at them linearly to an extent, but you also make sure that you look at them as a whole. And package design is a whole concept. It includes microbiology, it includes produce physiology, it includes the polymer, it includes how we're going to put it in the package, and are we going to automate it? And seasonality can change packaging needs. So, you know, if you're growing in the Salinas area versus growing in the deserts of Arizona or Southern California, your physiological properties are changing. So you need to adapt to that. If you're growing in a greenhouse versus growing outdoors, make no mistake that the physiological properties of that produce are different, and the packaging needs to compensate for that. And cannot stress enough that a thorough understanding of the requirements are needed to get the desired results. And if you look at your packaging company, they truly supply a technical support service that happens to include packaging. If all they're doing is they're giving you a package without the technical support, then ladies and gentlemen, they're doing you a disservice. So I'm going to turn it back to you, Joelle, for the final thoughts and comments.
Sure. Well, thanks, Jeff. So I think, you know, the recurring theme that we've really been trying to talk about during this process is that you've got to design optimization concurrently with all of these different variables. And those can start with the seed and the variety development, because obviously if they're not as vigorous of a plant going into your process, they certainly aren't going to get better later on. But we need to think about, you know, is not just, you know, what are the genetics of that plant and what are the susceptibility, but also how we're harvesting them. And as Jeff said, which area. So your plant is going to be in a different physiological state in different areas, whether it be the desert or whether it be in Salinas. The soil quality itself will impact how the plant grows, but also your product microbiology. So just like your plant behaving in different ways in different areas, so will the microbiology. And what's there and not there will impact your shelf life. Jeff talked a lot about how we measure the oxygen and CO2 in a package, because that's going to impact your product's shelf life. But also is that as you remove oxygen, it becomes more anaerobic. We certainly allow for the proliferation of organisms that thrive in microaerophilic environments or even anaerobic environments. Most of those may not be food safety issues, although some could be, but they certainly can cause some very significant changes to your product in terms of pH and flavor quality, and obviously just physical shelf life. Processing also has a lot of impact. And I think we often think with fresh produce is that we harvest it and we just throw it through the process and we'll bag it up and it will be fine. But the physical degradation, as well as the introduction of bacteria that can grow from your processing steps, whether it be your wash rooms or your dryers or your dicers and your slicers, all of those things are introducing new surfaces and environments that could impact your product's success. and your packaging needs, as to what Jeff has mentioned. So all in all, when going out, there's an enormous amount of different variables that you need to think about, both agricultural variables, as well as processing, as well as plant physiology, as well as microbiology. And the unique thing that you need to think about is not just recognizing that they need to be independently looked at, is that they actually need to be concurrently looked at. And they need to be looked at most likely at different stages and seasons in your crop. As your product moves to different areas, so too should your experimental design. And the partnership between Eurofins and QFresh is effectively combining all of these attributes so that instead of looking at these in isolation, we can look at them in tandem. So we can understand, is it the product that's changing, or is it the population of microbiology? Is it the nutrient degradation that we're looking for, or is it the package that's actually impacting the degradation of those nutrients? So all of these things are partnership opportunities, as well as critical pieces to designing your shelf life so that your product is going to have the most successful end life and ultimately a happy customer and consumer. So with that, we'll open it up for questions. We have our contact information here.
All right, thank you for that presentation, Joelle and Jeff. So I'm going to go ahead and just launch the poll as well here on the screen. I'd ask if anyone is interested in receiving follow-up after the webinar. But again, at this time, we will go ahead and please feel free to submit your questions in the Q&A section. So the first one that came in is, can you explain the UNIs such as OTR and all others?
So I'm sorry, Amy, say that again,
Yes, can you explain the UNIS such as OTR and all others?
Well, OTR stands for oxygen transmission rate, if that's the question.
Oh, yes,
oxygen.
And I think they're asking, can you explain the units? Okay, such as OTR and all others.
Okay, got it.
Sorry about that.
Oxygen, no worries. Oxygen transmission rate, depending if you're in North America or if you're in the rest of the world, is either in North America, it's cc's per 100 square inches per atmosphere per day. In the rest of the world, it's cc's per meter squared per atmosphere per day. And that's how you measure, and there's a significant difference between those two units. So when somebody lists the oxygen transmission rate of their packaging or their film or polymers or perforations, it's very important to understand whether or not you're talking about cc's per 100 square inches per atmosphere per day or cc's per meter squared, because if you're using the wrong limits with the number, it can quite quickly lead you astray.
Okay, wonderful. Thank you, Jeff. And again, please continue to submit your questions. Another one that came in here, is there a difference between industrial and backyard compostable?
Yes, I'll take that one again. Yes, there is a very significant difference, and it has to do with the temperature of the compost pile. In an industrial compostable and products that are industrial compostable, they compost under very high heat, much higher heat than you would see in a backyard compostable. So industrial compostable products will compost both in an industrial composter and a backyard composter. In a backyard compostable products will only compost in a backyard composter. They will not compost in an industrial composter because there's not enough heat. So in other words, if you create a package out of a polymer that is only industrial compostable, it will not compost in a backyard composter.
Great, thank you. Another one here, can seed selection impact package design?
Absolutely. I think as both Joelle and I mentioned, the seed variety can very significantly impact package design, especially in although it's very true in vegetables, it can be very true in fruit. Depending upon the variety or cultivar of fruit, we've seen in excess of 4 times a fourfold difference in respiration rate between the same fruit, but within different varieties or cultivars of that same fruit, a fourfold difference in respiration rate and physiological properties. So the seed variety makes a very significant difference. So if you're growing, let's say in the Salinas area, and you use one kind of seed variety, and you're growing in Arizona, you may be using another seed variety. And if you're growing indoors, you may be using a third seed variety, and they could, all 3 of them, potentially have different physiological properties.
And I'll just jump in on that too, is that I think, agriculturally, growers, like Jeff said, they pick different seeds for different areas because they know there's different performance in the field. Well, the people who design products and or the customers who are asking for products may be talking to seed developers in respect to, oh, we really like a purple color, or we really want this flavor. But they may not think about how that product may perform differently in different environments, as well as, thinking about just genetic variability. If you think about humans, is that, every person responds differently in different situations. And so too is true with seed development. So when you put that into a package and then you challenge it, which, is obviously what's happening, you've cut it, it's technically dead, it's living and breathing, it's now sealed up in a package, is that if you don't think about that when you're also designing the attributes you're looking for, then you will ultimately not have a happy experience at the end of your product, most likely. So it's just something to think about, is that we often think about seed characteristics and plant characteristics to get us some certain trait at the end, but we also need to think about how it's going to perform in the package. So, to Jeff's point, is that it's not something to think about at the very end. It's the thing to think about at the very beginning.
Wonderful, thank you. And it looks like we have another question here. Is gas flushing necessary on all fresh cut products?
No. Gas flushing is a very effective tool to get the oxygen level down below about 5% in order to minimize enzymatic browning reactions or pinking, which is very common in iceberg lettuce. It's common in cause romaine lettuce. It's common in butterhead. Those 3 varieties of leafy greens almost always use gas flushing. In other products where enzymatic browning or pinking is not an issue on the produce, there's no need to gas flush. Early on in the development of the fresh cut produce industry, it was thought that gas flushing helped everything. It doesn't necessarily help everything. Only if you need to prevent those enzymatic browning reactions. And gas flushing can be detrimental, because if your package is not designed properly and you gas flush, then you run the risk, a much higher risk, of going anaerobic, which can cause those off flavors and off odors. So gas flushing is a very effective tool, but should only be used when needed.
Okay, thank you for that answer, Jeff. And it looks like if anyone has any questions, I have been monitoring the Q&A, and I don't see any other ones coming through, but I'll just give everyone just another minute if there's anything else that you would like to ask the presenters today.
So while we're waiting for that, Amy, I just want to go back to the compostable industrial versus backyard and just make sure I stated that clearly. So if you have a package that is industrial compostable, it will only compost in an industrial composter. It will not compost in the backyard. If you have a backyard compostable package, it will compost under either situation. I just want to make sure I clearly stated that.
Okay, thank you for that clarification. And it looks like I do not see anything else coming in. So I think we will wrap up our Q&A session. Again, just a couple of items. I want to remind everyone that a recording of this webinar, along with a copy of the slides that were presented today, will be available within 3 business days. And again, I want to thank everyone for attending and for this very informative presentation today. Thank you again, and that will conclude this session.